Hello and welcome. My name's Matt Gollock. I am the program lead for the Aquatic Species and Policy Program here at ZSL. Welcome to you all to the sixth Science and Conservation event of our 2023-24 program. We've got three excellent speakers lined up, and then we'll have time for Q&A at the end. So without further ado, I'm going to introduce our first speaker, who is Hannah McCormick. Hannah is the project manager in the Estuary and Wetlands team at ZSL. Our work focuses on UK aquatic species conservation, including marine mammals and sturgeon. She's a member of the UK Sturgeon Alliance, of which ZSL are founding members. The UK Sturgeon Alliance is a group of charities that champion native sturgeon conservation, and last year Hannah worked in collaboration with the Alliance to publish the UK Sturgeon Conservation Strategy and Action Plan. The Alliance and ZSL sturgeon work now focus on delivering the action plan, and these activities include habitat restoration for sturgeon, bycatch mitigation, and understanding past species distributions. And with that, I'm going to hand over to Hannah.
Thank you all so much for joining tonight. For our speakers who will be following me, for Matt for chairing, and Harriet for organizing this. Really excited to be here talking to you about sturgeon. So I'm a project manager, as Matt mentioned, at ZSL, and I'll be speaking to you about ZSL's work in close partnership as members of the UK Sturgeon Alliance on restoring the UK's native sturgeons to UK waters. As a quick outline of what I'll be talking about, first I'll give you an introduction to the royal sturgeon. I'll talk a little bit about the historical context of sturgeon in UK waters. I'll then go on to talk about some of the restoration activities that are turning the tides for sturgeon in Europe. And I'll finish on a really important topic, increasing survivability at sea. So diving right in, let's meet the royal sturgeon. As a very rare species, it is one that a lot of people don't know too much about. One thing to know about it is that it does have quite a few nicknames, including the royal fish. So we'll talk a bit more about that one when we come to the historical context section. But to describe a few of its other common nicknames that it's often goes by, firstly, the dinosaur fish. So apart from looking quite prehistoric, sturgeon are often referred to as dinosaur fish because their ancestors were around at the time of the dinosaurs. So they're often called living fossils. Sturgeon are a family of 27 species, one of which was declared extinct in 2022, the Chinese paddlefish. They're the most critically endangered group of animals on the IUCN Red List. We have two species of sturgeon that are native to UK waters. That is the European sturgeon, which is critically endangered globally, and the Atlantic or Baltic sturgeon, which is vulnerable globally. Both of these have experienced massive declines across Europe, and neither of them currently have spawning populations in Europe. Another common nickname that sturgeon are referred to as is river monsters. This one is slightly misleading. I say slightly because they can grow over 5 meters long. They can live over 60 years old, and they mature at 10 to 20 years. So 5 meters is massive and could be described as quite monstrous. However, they are bottom feeders that feed on worms, crustaceans with their Hoover-like mouth that doesn't have any teeth. So in this way, they don't quite fit that description. They are migratory fish, fitting tonight's theme, and they spawn in fresh water on gravels and stones. They then make their way down towards the estuary, spend a few years in brackish waters, getting used to salt water, and then they will migrate out to coastal and marine waters, where they spend the majority of their later juvenile and adult lives returning to fresh waters to spawn. Now on to the historical context of sturgeon in UK waters. So, firstly, starting off with an explanation of the royal fish. So sturgeon were declared royal fish in 1324 by Edward II. That's 700 years ago, exactly. And that status means that any sturgeon landed in UK waters has to be offered to the crown. The status still stands today, but is more of a formality because of the protections around these species and just the fact that they're very rarely landed. The last sturgeon to be accepted by the crown was in 1980s. So this kind of was our first indication of sturgeon being quite significant in British culture. But if they're no longer here and they've been gone for quite a while, how do we know that they were in UK waters? Well, there's loads of information from lots of different historical sources, including newspapers, museums, books, landings records, and a massive effort has been made by Steve Colclough of the IFM, who's also a very important member and founding member of the UK Sturgeon Alliance. And he's put together a massive database of thousands of these records, which really shows us the distribution of sturgeon across our freshwaters, estuarine, marine, coastal, and it's really given us a baseline for the historical presence that we didn't previously know much about. It's also given indications of signs of spawning in our freshwaters, which again, until this database was put together, we didn't really have any indicators of. So specimens like this one, juvenile specimen from the Museum of Zoology in Cambridge, indicates that spawning likely took place in UK freshwaters. Some of the historical accounts of sturgeon in freshwaters, what it was like to see a sturgeon in your local river, are absolutely incredible. This one from the River Frome in 1911 describes the experience these men had while fishing for salmon when a sturgeon came in the center of the pool, appeared a huge back fin shaped like that of a shark, with a great back, scales like armor plating, and repeatedly the monster rolled on top of the water close between our feet. Unfortunately, this fish, like most sturgeon that wound up in, or at least were spotted and found in UK rivers at the time, was fished out and ended up being offered to the crown. This one was refused, and it was suggested that instead it be displayed in the local museum, which it was and still is in the Dorset Museum and Art Gallery. Unfortunately, the popularity of this fishery, fish that was popular for its meat and its eggs, which we know as caviar, meant that with the intense efforts of steam trawlers, that they were massively overfished from the 1880s onwards. Other threats also contributed to the decline of sturgeon across Europe, like barriers to migration that interrupted spawning, and also habitat destruction, but intense fishing was definitely the main contributor to the declines of sturgeon across Europe. One example of this is Skipper Newson of Grimsby, who was nicknamed the Sturgeon Hunter out of the northeast of England. And on one of his voyages in 1907, over 4 days, he landed 285 sturgeon, ranging in size from 3 feet all the way up to over 10 feet long. So it really was massive, huge, intense efforts. And because of this, by 1950, sturgeon landings were unusual. That was the case across most of Europe, most of Western Europe for these two species. And because of their late maturity, it means that it takes longer for these species to recover. So they weren't able to keep up with the intensity at which they were fished. And by 2000, there were no wild spawning populations of either species remaining in Europe. But this isn't a sad story. So the tides are turning for sturgeon, and the European recovery efforts have gave them a new hope for recovery for both European and Atlantic sturgeon in Europe. Ex situ breeding programs for both species started in the early 2000s and have been ongoing since. 1.6 million European sturgeon have been stocked and 4.5 million Atlantic sturgeon. Again, because of their late maturity, it takes a long time for spawning to result from these reintroductions. So that hasn't been seen yet, but early indicators of success have been observed, like fish returning to the rivers from which they were released. In response to these massive European efforts, and with our newfound knowledge that this incredible historical database has given us about past UK distributions, the UK Sturgeon Alliance was formed in 2019, and that was a group of charities coming together to champion UK native sturgeon recovery. That was with ZSL, the IFM, Blue Marine Foundation, Severn Rivers Trust, among a few others as well. And last year, we published the UK Sturgeon Conservation Strategy and Action Plan. And this kind of gives us the roadmap towards recovering native sturgeon species in the UK. And that QR code will give you access to the document if you're interested to read a little bit more. I don't have time to go through everything in there, but I am going to focus the last bit of this talk on a really important goal that's laid out in that plan, which is increasing survivability of sturgeon at sea. So one of the threats to sturgeon at sea is accidental capture. Because of their protected status and very endangered status across Europe, fisheries no longer remain for these fish. But they do sometimes accidentally get caught up in gear that's targeting other fish. And this is often called bycatch. Luckily for sturgeon, they are quite hardy fish. And most of the time when this does happen and they're pulled on board, they are still alive. And this means that the response of the fishers who have accidentally caught them is really important and could improve their chances of surviving. And for this reason, sharing information about sturgeon and their endangered status across Western Europe is absolutely critical. These efforts to share information and to work closely with fishers have been ongoing in many countries across their range, including Germany, France, and the information that fishers have of when this happens is really critical to monitoring the reintroduction efforts. Because of the huge range of these fish, they really can roam really far. Monitoring for them is an absolute nightmare. So these incidental events provide really, really valuable information about where they are and how many there are. Their range also shows the importance of collaboration between European nations on their recovery. As increasing numbers of sturgeon are caught in UK waters from these reintroductions, it definitely emphasizes the importance of the UK getting involved and being part of this collaborative effort. So these are recent efforts and sort of plans laid out for how the UK plans to get more involved, but collaboration with our European colleagues has been absolutely enlightening because they have huge insights, like, for example, tagged fish are way more likely to be reported than untagged sturgeon. So related to this, again, along with working closely with our European collaborators, we also plan and know the importance of working closely with UK fishers. The main focus will be to run these information campaigns that have been ongoing in other countries, and focusing those in the UK to raise awareness of these species and their incredibly endangered status across Europe. We also plan to co-develop solutions with fishers to allow for easy and safe reporting and to protect any sensitive information, make sure that data is being used in the way that it's intended, and also to help share and also get new insights about ways for a quick and a safe release of sturgeon that are caught. For example, keeping them in the shade, keeping them cool and wet when they are on board, and then releasing them as quickly as possible. And using this fisher knowledge is absolutely critical and will massively contribute to the success of our efforts. So this is the type of information that we need to make sure gets out there, and we will continue to share in different networks and through different channels. So recording the length, the weight, condition, date, and location is all very important information, and a photo helps us identify species. If the fish has a tag, make sure that is left on and write down the number. Because it's a protected species, even though in the UK it is a royal fish, it still needs to be released, no matter its state. If it's alive, it should be released as quickly as possible, but it still needs to be released even if it's dead. And the information should be reported to the relevant authority depending on where it's caught. That's the MMO in England, Marine Scotland, etc., and to the receiver of the wreck, and then to the UK Sturgeon Alliance, to help us understand that data and share it with our European collaborators and track the success of bringing these fish back to our coastal marine and hopefully eventually freshwaters. And if a sturgeon does get returned safely like this one, then we are doing our part to contribute to the recovery of these fascinating species across Europe. So the main takeaway message is to return it and to report it. And finally, in conclusion, just to summarize, the historical evidence that's been gathered about UK sturgeon distributions shows widespread across UK freshwaters, estuarine, coastal, and marine. And the UK Sturgeon Alliance supports the European recovery efforts with our ongoing conservation strategy and action plan. And finally, if a sturgeon is caught at sea, it should be returned and reported no matter where it is caught. And just to end on a bit of an inspirational note, sturgeon have really already started to capture the fascination and the imagination of the younger generation, like the student who made this pledge here. And their recovery could be a fantastic example to show young people that species that have been lost to our living memory, species absolutely on the brink, incredible species like 3-meter dinosaur fish can recover with our help and efforts. But there's lots of people to thank in relation to this work, including some of our European collaborators, our UK Sturgeon Alliance partners, our funders, Garfield Weston, Natural England, and many, many more. And thank you to all of you for being here and for listening.
That's great. Thanks so much, Hannah. Excellent start to our list of speakers. I will let you go for now, and we'll come back for some questions later on. So now to our second speaker. I'm very pleased to be able to introduce Dr. Caroline Durif, who I have the good fortune to work with on occasion through our work on eels. Caroline is the principal research scientist at the Institute of Marine Research in Norway. She received her PhD in France on the migration of the European eel and its challenges linked to hydropower. Her research now deals broadly with fish migration and life history strategies of fish. She also specializes in studying how the behavior of marine organisms can be affected by anthropogenic effects, but also related to the development of human activities in the ocean, such as renewable marine energy and offshore surveys. Caroline gives scientific advice on the status of eel in Norway and at an international level. She chairs the working group on eel under the umbrella of the International Council for the Exploration of Sea, the European Inland Fisheries and Aquaculture Advisory Commission, and the General Fisheries Commission for the Mediterranean. So over to you, Caroline.
Thank you, Matt. So, well, I'm going to talk about another mysterious fish, if not the most mysterious fish, the European eel. And the story starts more than 2,000 years ago with Aristotle. Aristotle was the first person to work on biology systematically. So he was collecting samples, observing nature, and pretty much in the same way that we are doing it now. And he studied reproduction in many fish species quite accurately. But when it came to the European eel, or to eels, he was perplexed because he couldn't find any sexual organs, what today we call gonads. And for him, the eel was neither male nor female and could engender nothing. He also couldn't find any eggs in eels, such as those we find, for example, in sturgeon or in salmon. And yet he saw these tiny, transparent, spaghetti-like glass eels coming out of the seaweed. And therefore, for him, eel was the typical example of spontaneous generation. So in his own words, eels were derived from the so-called earth guts that grow spontaneously in mud and in humid ground. In fact, eels have at times been seen to emerge out of such earthworms. And so the mystery persisted for hundreds of years. And during the 18th century, the eel was thought to be viviparous, meaning that it bared its young so that they hatched. They thought that eel came from an egg like all fishes, but the egg most often hatched inside the belly of the mother. Now, why did they think this? Because this was following the pioneering work on microbiology with the development of the microscope. So Antonia van Leeuwenhoek who saw these tiny black worms inside the eel, inside the bladder, that seemed like young ones ready to hatch. Now, you probably guessed already that these were not eel larvae, but parasites. The actual eel larva looks like this. So it doesn't look like an eel. It looks like a leaf, and that's where it gets its name, Leptocephalus larva. And it was actually initially thought to be a completely different species, which they called Leptocephalus brevirostris. And it was only 40 years later that they made the connection between the Leptocephalus and Anguilla anguilla. And this was done by two Italian biologists who kept a Leptocephalus in an aquarium and witnessed the metamorphosis inside the aquarium to Anguilla anguilla. So problem solved, or half solved, and they concluded that the eel was spawning in the sea because they had found these larvae had been found in the Strait of Messina in the Mediterranean in Italy. So the search for the spawning area of the eel started, and it was Johannes Schmidt, so a Danish oceanographer, who surveyed the Atlantic Ocean and had and trawled for over a period of 20 years. And he counted and measured hundreds of Leptocephalus larvae and mapped their geographic distribution. and he found that the smallest ones were located in the Sargasso Sea area. And therefore, he concluded that that's probably where the European eel was spawning. So since then, many scientists have gone to the Sargasso Sea and trawled the Atlantic and collected leptocephali. And everybody came to the same conclusion that the smallest larvae were in the Sargasso Sea, and that's also where the concentrations were highest. So incredibly enough, this wasn't before 3 years ago that the spawning area was actually confirmed by colleagues, among which Matthew Gollock, who they tagged, this was after years of efforts of tagging eels, which so in the beginning, the eels were released from the European continent, but the problem is that these eels are tagged with what we call pop-up satellite archival tags. And these are sensors that are connected to a balloon, and the sensors record the data necessary for locating the eel. And after a certain period of time, the balloon floats back up to the surface and transmits the data to a satellite. And the problem is that this balloon makes the eel a little bit vulnerable to oceanic predation, and they get eaten by whales and sharks. And so the idea they had was to catch eels as close as possible to the spawning area in the Sargasso Sea, and this is what they did. And caught eels in the Azores and tagged them, and finally they were able to track them down to the Sargasso Sea, and the spawning area was confirmed. However, we have never found any naturally spawning adults there. So this is still sort of a mystery. So this is the life cycle as we know it today. The leptocephali hatch in the Sargasso Sea, and then they drift with the Gulf Stream to the European continent and North Africa and the Mediterranean. And when they reach the continental shelf, they metamorphose into glass eels. And then these glass eels become pigmented. We call them elvers, and they colonize freshwater systems and stay there for a certain extremely variable amount of time, depending on where they are. And at this stage, we call them yellow eel. And then after their growth period, the yellow eels transform again into a typically marine fish, marine-looking fish with its black back and a silver white belly. And this is the silver eel that will eventually migrate back to the Sargasso Sea to spawn and die. So they only reproduce once in their lifetime. So the incredible information also that we know about eels is that all of the eel, as you can see, they're widely distributed from northern Norway down to Morocco, and then far into the Mediterranean. And all of them spawn in the Sargasso Sea, meaning that they all have the same genetic material. And despite having the same genetic material, they can withstand very different conditions, like temperature conditions. They can live in habitats with freezing waters. They can live in rivers that are almost drying up. And they also can colonize different salinity environments. So typically you will find them in rivers and lakes, but also in brackish water, in marshes. And some of them decide to stay in the marine environment, so in coastal marine areas. And we don't really know why, what drives their choice of habitat. So this is still something to work on for us. But what we know is that no matter what, they will try to go up freshwater systems, because it seems that they get very important lipids for their reproduction, which they get mostly in freshwater systems. And so this shows a picture of elvers trying to climb up no matter what. They're quite incredible. That's why you will find them inland, maybe thousands of kilometers upstream sometimes. And but this is good that they could spread everywhere, but it also makes them extremely vulnerable to hydropower. So at the time of their migration, usually in the fall, they all go down, swim downstream rivers, and can get caught up in turbines. So there are solutions today called fish-friendly turbine, but they're not very efficient. And also, even if the eels do not get chopped up but are simply injured, it will decrease their chances of swimming. For some of them, they have to swim 8,000 kilometers to reach their spawning area. So this is one of the reasons why the European eel is critically endangered, since it's classified as such since 2008. And the way we measure, we evaluate the population is through what we call the index of juvenile recruitment, which means basically the number of glass seals swimming up freshwater systems. And today, we are actually at between 95 to 99% of what the level was before 1980. And as you can see on this figure, it's also the case for the Japanese eel and the American eel, which are also called freshwater eels. Another cause is clearly overfishing, because the eel is caught with many different types of fishing gear. And it is, many people don't like eel, but it's actually quite tasty. And it's a very fatty fish, and it was a very important food item for Native Americans. And it was actually the first meal shared between Native Americans and Pilgrims. So it shouldn't be turkey on the Thanksgiving table, but actually eels. Since they migrate during the fall, then it was very a nice food item to have just before the winter. It's not only silver eels that are consumed, and here I show the life cycle. And as you can see, we consume every life stage. So even the glass eels and the yellow eels, there's sushi, and the silver eels, which is usually smoked because it's very fat, as it's the eel that's going to reproduce. Now, it's not only humans who like to eat eels. It's also, and this is believed to be one of the threats, although it's natural mortality, as we call it. But it's the explosion of the population of cormorants has probably also impacted most of the population. One probably very important cause of the decline is linked to habitat loss. So of course, dams, and which prevent maybe upstream migration for some of them, but also all of the drying up of the wetlands, which are a typical and very important habitats for eels and many other amphibians, for example, which have also suffered from the same kind of decrease. So I've talked about some of the threats, not all of them, and this is why, even though this species was quite successful throughout the millions of years, but today it's really endangered, and pollution is also a problem. The eel reproduces only once in its lifetime, so it's not able to evacuate the contaminants. So, and also these contaminants which accumulate throughout the eel's life, they are transmitted probably to the larvae and causing deformities and reducing the recruitment. There's also climate change related causes due to increasing temperature in the Sargasso Sea, which decrease the primary production, in other words, food for the larvae. And then finally, there's this parasite again, which has been infecting more than 80% of the eels. and it sits in the swim bladder of eels, and it also decreases their chances of reaching the Sargasso Sea. So I'm going to finish or show you at least these two extreme examples of eels, like I said, with the same genetic material, who are able to adapt to very different types of environment, from the North Cape in Norway, where they reach they have a long life cycle, up to 50 years old, maybe more. They have a slow growth. They grow to be almost 1 meter long, versus you have eels in the southern part of the distribution who have a shorter life cycle, 2 to 6 years old, and who are shorter, so less eggs. But I think it's probably due to this incredible capacity to adapt to different environments and to show diversity within the same species that the eel has been able to be successful. And unfortunately, it's really reaching now a critical moment for eel. And so with my colleague, Jan Dag Polman, we chair the working group on eels, which gives advice for the European eel. And it's basically, we meet every year, and we collect, we share the data. It's all experts on eel from Europe and countries around the Mediterranean. We share the data, we write a report, we try to find ways for eels to recover, and so it's a nice example of diversity at a smaller scale. And if you're not convinced that eel is a really cool fish, here are some examples throughout the literature and cinema and everything. And yes, please contact me for any questions. Thank you.
Thanks very much, Caroline. Yes, please put questions into the Q&A, and Caroline will come back to you at the end for the panel to ask those. She completed her PhD in French Guiana on the ecology and biology of Goliath grouper in French Guiana. She joined the Game and Wildlife and Conservation Trust to track the migration of salmon and sea trout as juveniles and adults. This work was part of a European project named Salmonids Management Round the Channel. She's presenting tonight the results of the marine migration of sea trout. So over to you, Céline.
Thanks, Matt. All right. So I would like to thank you guys for inviting me and share the work we did on sea trout. And so it's another migrating species, and with other questions, but you can see that it will be very similar to the presentation before. So I will talk about the work we did to found the migration of sea trout in the English Channel, and I will highlight our findings and how it can help the managers to protect the species. So this work has been done in a European project between England and France with different labs as collaborators. And I put all the institute logo in here to thank all the colleagues that helped with this work. So first, I just wanted to summarize what is important information when we want to manage a species. And the first information is to know the life cycle. So you saw the prior presenters that talk about how they manage to find the life cycle where they reproduce. So it's the same for every species, and we need to have information not only on the reproduction, but on where they are living and where it's best for them to grow and to be big and to reproduce a bit more, what's the structure of the population of the species, and what is impacting their survival. So all kind of information that you see in the presentations. And when we've got all this information, then we can adapt the management, and we can manage the species efficiently. When we have a lack of information, then it's a bit more difficult, and sometimes the management is not appropriate, and we are not managing very well, and it's not good enough for the population. And it's even worse when we are trying to understand what is impacting the life cycle of the species, whatever it is. It can be invasive species, it can be fishing, it can be pollution. And if we do not understand our impact on the species, then obviously the management is even less efficient, and it's not good. So our work in the project I'm presenting was to try to find the evidence that was necessary to improve the management for the sea trout. So sea trout are migratory species that are reproducing in rivers this time. They are growing in river, and they have like different names for their different life stages. So when they are very little, they are known as spar, and then they grow a bit more, and they are known as smolt. It's the time to migrate to sea. So as smolt, they migrate to sea where they can grow and find food to grow better. And when they are big enough, they can have like their sexual maturity, and they go back to the rivers to reproduce. So because rivers are accessible, we have information on the life cycle of the sea trout when they are in fresh water. And there is some specific rivers like the Frome that was already cited previously, where the populations are surveyed, and we can know if the population are increasing or decreasing. And around Europe, the general observation is that there is less and less individuals that are coming back from their migration at sea, and so the population are decreasing. Obviously, we have much less information on what is happening outside of freshwater, so when the species is in the marine environment. And we don't have so much information on why this mortality is increasing at sea. And that was the goal of the SAMARCH project, to understand what is happening at sea and where the sea trout are going, to try to improve the management. So on this project, we focus on 3 different rivers in England and in France. So you've got the Tamar in south of England and the Frome, and as well as the Brel, that is a river in north of France. Our objective during this project was to understand where the sea trout are going and when they are in the English Channel. We were trying to identify the migration routes, and if we could, the feeding areas. If they stop somewhere to feed, then it would be necessary to identify this area to focus the management when they are at sea. So if we talk a bit more about the different sites, it will be important. And we've got the Tamar, that is a very long river that is ending in a nice bay of Plymouth. And it's quite a natural river with no obstacles for the trout to go at sea. Whereas on the Frome, it's ending with an estuary with a shape of a ball, and there is a very tiny exit for the fish to find the exit. And the Brel is a very anthropogenized harbor, because you've got no exit for the fish. Everything is closed by gates, and there is just a fish path somewhere that the fish has to find in order to reach the sea. It's all kind of difficulties that fish has to cross before to reach the sea, and we studied the survival in the estuaries, but I will focus on the marine part tonight. So how we followed the sea trouts and we tagged them. We didn't tag them with a pop-up tag as Caroline explained, but we used other kind of tags that I will explain how they work. But as the fish are going at sea, they need to be identified by fishermen or people that catch the fish. And so for that, we did a tattoo on the belly of the fish, and we put a plastic tag on the dorsal fin just to externally visualize that the fish was followed by scientists. And the other tags, these ones, like the orange one that is called the data storage tag and the black one that is an acoustic tag, were inserted internally of the fish. So obviously you can't know that these tags are inside the fish when you capture it. Also, we did put also a different kind of tag that is named a pit tag. It's what is used in freshwater to count the population and to survey how many fish come back from their migration and things like that. So the orange tag is called the data storage tag. The DST is recording the temperature. Internally, the fish is not regulating its temperature. So the temperature recorded by the tag is reflecting the temperature of the water. And every 2 minutes, this tag is recording the temperature as well as the pressure. So we know the pressure at which the fish is at a specific moment. However, this tag is not sending any data. So it means that we have to recapture the fish to get the tag and the data and to know the migration routes of the fish. So to do that, in fact, the tag is working with the pressure and the temperature. So when we look at the pressure, then we can know that the fish was going down, the pressure is increasing with depth. So we can know that the fish is having vertical movement. And for horizontal movement, what is interesting is the temperature. So if this fish is trying to go in warmer water, then we will have the temperature, we will have the information, and we will compare the temperature with a map of ocean water temperature around the world. So we did this comparison, but for that we need the information of when the fish arrived in the marine environment. And that's why we use the other tag that is an acoustic tag. And this tag is transmitting the code every 30 seconds, is transmitting a code that is telling the number of the fish. And this code is received by this black bottle that we deployed on the little dots that you can see in this map of the Frome River. And so when the fish with this tag is going at sea, then we've got the information on all these little dots that the fish was there at the specific time. What is important for this talk is the last receiver that has the information of the fish arrived in the marine environment. And for us, that's the starting point. The fish is at sea and we are trying to follow its migration route. But then we have to recapture it because the orange tag is not transmitting any data. So for that, we are waiting for the fish to come back from its migration. And then it crosses a receiver that is transmitting us a text. The fish is there. And then we electrofish the fish to recapture it and get our data back. So thanks to all this information, then we can try to geolocate the fish. It's a kind of an estimation of where the fish was. And so we've got these results. So here you've got like a map of where the sea trout we tagged went in the English Channel. You've got in yellow the fish that we tag on the French river. On green, you've got the fish that we tagged on the Frome River in the middle of South England, and in blue, the one from the Tamar. So you can see that sea trout are going quite far away from their river sometimes, and that was really interesting. But what is a better information to manage the species is on this map, and here you've got the color that is representing a number of individuals. So when you are in red, orange, or yellow color, there is a lot of fish that are on this location. So this map is telling us that there is main migratory corridors for the sea trouts that we followed. And we found like 2 or 3 main corridors, and that's the moment where fish are kind of, most of the fish of the population are in the same location. So if we have to protect the species, that will be the location where it will be important to protect the species or regulate the species at this location. And then what we found, so here you've got an example of the migration routes of different fish, like the Brell fish, the Frome fish, and the Tamar fish. And another information that you can see that the French fish is going towards the English waters, and that the opposite, the English fish, either the Frome or the Tamar, are using the French waters. So it means that we need to collaborate between countries to be able to do a common strategy to try to improve the management of the species. Other information that we found, and it's really scientific, but you will see everything is really helpful for the management. So here I'm presenting graphs. So you've got in yellow the fish from the Brell, in green the ones from the Frome, and in blue the Tamar. It's always the same color code. And here you've got the distance at which the fish went, depending on their home river, so the starting point. And you can see that if you look at the shape of the graph, it's just that the fish are the furthest away in the middle of the migration. So here you've got the weeks of migration. So you've got the 3 first week, and then we are in the middle of the migration, and then you've got the 3 last week of the migration. So the fish is further away in the middle of the migration, and then it comes back. So it's just saying that the fish is migrating with a loop shape approximately. And then the other information that is important is that the Brell fish are going further away from their home river than the Frome and the Tamar fish. They are more adventurous fish, and it means that the French people that want to manage their population, they have to collaborate with other countries where these fish are going. Another interesting information is what is the distance they are traveling every day during this migration. And there you can see, so here you've got the distance that they are traveling daily. And then you can see that it's an opposite shape. The fish is migrating faster, longer every day at the beginning of the migration and at the end. Whereas in the middle, they are not moving so much. They are kind of wandering around. And if we look a bit closer to the fish, and we took an example, then we try to understand, and we look at fish that went for this migration for the first time or the second time, and then the 4th and the 5th time. And you can see that the shape of this daily distance they are traveling are stronger and stronger, more the fish did a prior migration before. So for us, it can mean that at the beginning, when the fish is having not so much migratory experience, the fish is looking for its way, is looking for the food at sea, but is not knowing where the food is. Whereas when it's several times that it's doing the migration, then maybe the fish got a purpose, he knows where the prey are, and then he's going there faster to reach this area fast and to feed in this area much longer during the migration. So that's quite important, mostly because the fish will be away from the shore, maybe for a longer period of time. And it's important because managers are different if the fish is at the coast or if it's further away from the coast. So here, for example, below this red line, with the distance to shore, below this red line, that's the environment agency that is managing the waters. And further offshore of this distance, then it will be other people like the MMO. And then just on this graph, what I want to show is that the yellow lines are all the time during the migration, offshore or more coastal. It just means that sea trout, some of them stay coastal and others are going offshore. And when we looked at which fish it can be, then we have this tendency again with the number of migration that the fish did before. And younger fish, the fish that has less migratory experience, stay coastal, and the ones that are bigger and that have more migratory experience are going further away at sea. So this is important because if we want to protect the small one or the big ones, then we need to manage the location where they are, and if they are in different locations, that's important. So other information that was very nice, mostly for managers and for the fishing rules, and that was the diving behavior of the fish. And if we look at the blue graph to begin with, then in vertical, in y-axis, you've got the depth, so the pressure the fish went at. And here in x-axis, then you've got the 24 hours of the day. So here is the representation of the diving behavior of one day in a specific fish. In blue, the tailor, green, the foam, and yellow, the braid. And you can see that the pattern of diving is changing through the population. You've got tailor fish that are very close to the surface at night. And when the daylight starts, then the fish start to dive and is going deep, but sometimes going back to the surface. The foam fish, it's quite the same behavior, but they are coming back to the surface more regularly than the tamer fish. Whereas if you look at the braid fish, then they are diving all the time, even at night sometimes, and they are doing a very important number of dives per day. So this information was important because it was believed that the sea trout were a surface swimmer, and fishing management was to protect the 3 first meters of the water column to try to avoid to capture sea trout and salmon. But with this information, we can clearly see that the sea trout are not only surface swimmers. Sometimes it's at night only for some population, and for others, and it's not even only during the night. So we need to be careful at the whole water column if we want to try to improve the population. So an important, and I will try to be simple on this one, but we try to find why sea trout were dying at sea. So it's kind of some clues that we have to follow to understand. And here I'm showing you like the minimal depth in white the fish went during their migration. So here it's a fish that was tagged on the 15th of January, and then he died in February. And what we can see is that the minimal depth in white and the maximum depth in blue were kind of different at the beginning, and suddenly everything is dropping, meaning that the fish sank at the bottom of the water, and at some point the fish was degrading, and then the tag was released when the fish disappeared, and the tag was at the surface at this time. So for this kind of things, and we didn't know what happened before, why the fish died, but the fish sank, obviously was not good. And this explains like 30% of our mortality. So we don't know, maybe natural deaths, exhaustion, we are not sure. But then we have 20% of the fish that were predated by birds, and this we can see it looking at the pressure. So it was the blue color. So the fish was diving, the usual behavior, and suddenly the fish is out of water. We've got negative pressure. It means the fish was flying. And if you look at the temperature, then the temperature is suddenly increasing around 40 degrees, and it's the temperature of a bird. So it means that the fish was predated by a marine bird, and it was like 20% of our sampling. 10% of our fish were predated by mammals. We know that because the temperature is about 38 degrees, and there were a lot of diving behavior, but sometimes it reached the ground. So it can be mammals that is also going out of water. And then we've got 27% of others. Others, we can't really know what it is, but it's an animal that is not regulating its temperature. It's the temperature was not changing so much, but it was capable of diving quite regularly and a lot. So it might be sharks. And finally, then we had like a netting event as well. So the fish was diving, always the blue color, the fish was diving, and suddenly, like it was not diving, but we were following the tides. So the fish was caught in a net. And so the temperature stayed the same. And when it reached, when it was landed on a boat, then the temperature was increasing. So it was kind of different clues to understand what happened with the fish. And we managed to understand a bit better. So a lot of natural predation and some kind of fishing as well. So to conclude, with this project, we learned a lot about the ecology of the species. We didn't know where the sea trout were going at sea, so it was really a valuable project for that. And we didn't know the behavior or the roots of the fish, and now it's information that are known, and we can use them to manage the fish. And also lots of beliefs that change with this project. We believe the fish was coastal, surface swimmer, and were doing short migration. And that's not the case. They are going up to 100 kilometers offshore from the coast. And they are doing regular dives up to 80 meters. And they can be found more than 1,000 kilometers from their home river. So that's not what is can be done by eels, for example, but still that's not a short migration. What is important is that the sea trouts, you saw the graphs, they are crossing the border, so we need international management if we want to improve the status of the species. And then we identify very specific corridors that the fish are all taking when they are going at sea. So it's important areas where we need to focus our management, and then we need to talk with different managers and like the Environment Institute or the MMO, because they all manage different locations at sea. So we need to work all together to be efficient. And also, obviously, at the moment, there is some fishing regulation, but that are not appropriate for sea trout. So if we want to manage the species a bit better, maybe we need to change this fishing regulation. So thank you for your attention, and I hope all the talks were interesting for you. I'm staying there to answer the question.
Thank you, Céline. If I could ask you to keep your camera on, and for Caroline and Hannah to join us again. We've had some questions coming in on from the audience, so I'm going to start working through these. I've got a couple of questions of my own, but I'll keep them for later. So I'm going to start with Hannah. There's a couple come in. One question is, what's the main difference between the two sturgeon species? And just to follow that up, someone's asking if there's any examples of the large 5-meter sturgeon in museum collections in the UK.
The two species that are native to UK waters are very closely related and very similar in a lot of ways. So much so that they were believed to be the same species until about 1997. So in 1997, the Baltic sturgeon, which we now know, whose range was the Baltic region, is the same species that's in North America, the Atlantic sturgeon. And that's distinct from the European sturgeon, whose range is from the East Atlantic and Bay of Biscay, English Channel, North Sea region. So the UK kind of falls into the overlap of where the Baltic sturgeon can sort of roam from the Baltic, and we have evidence of both of them in our coastal and freshwaters. So it does make it tricky for disentangling which species was where, because all of the historical records are recorded as European sturgeon. So it will take a lot of archaeological and DNA analysis to untangle that mystery, as well as when we have photos from historical records, that can be a way to identify, but that's work we haven't done yet. But they have some slight morphological differences, but they do have very similar life cycles, and are very similar in a lot of ways, despite some slight differences in some of their habitat requirements and preferred prey, but overall very, very similar species. Oh,
Great,
and then the
thank
second
you.
question,
Yeah.
the 5-meter long sturgeon specimens. There is, I think, a 3-meter long sturgeon specimen in the London Natural History Museum. I don't know of any 5-meter long ones in the UK, and if anyone finds one, please let me know. But there is also, as well as a 3-meter long Atlantic sturgeon called Stanley the sturgeon at the London Natural History Museum. I don't think he's on display. I think he's in the tank room. So I think it requires a special behind the scenes tank room visit if you're interested in seeing it. But in that tank room, there's also a massive sturgeon head, which must have been over a 5-meter sturgeon. It's not one of our native species, but just seeing the size of this sturgeon head is really incredible. So if you are interested, I very much encourage you to book a visit to the Natural History Museum tank room.
Great, thank you. Next question relates to eels. Someone's asking about the Japanese and American eels and asks whether they have similar or concentrated spawning grounds.
Yeah, so the American eel is very closely related to the European eel. They spawn in the same location, actually, and except that their larvae stop. They take the Gulf Stream as well, but they stop to colonize the American continent. And the Japanese eel has a very similar life cycle in and also has very mysterious spawning area, which is also has never been found, although eggs have been found. And it's not as long as the migration of the European eel. And maybe actually, Matthew, you have the numbers and the distance, but it's quite long as well, and it's probably a deep spawning area.
Great, thank you. Céline, there's one here for you on trout. Do we have stomach info to tell us the different trout populations are following different food preferences in their migration?
So unfortunately, we don't have the stomach contents because they are digesting the prey during the migration, and when they come back, all the stomach were empty. So at some point where they come back in the freshwater, then stop eating, and it will not reflect what they are eating at sea, but what they were eating in the estuary anyway. So we do have mussel sample to study stable isotopes and to try to investigate what they were eating at sea. It's a work that we can do, but we don't have information yet.
Thank you. There's a couple of questions here I wanted to put to all of you relating to the threats. So someone's asked, it's interesting that eels can survive in such a wide range of environmental conditions. Do you think that means eels are more adaptable to climate change than other migratory species? And so to the other speakers, is climate change, a big issue for both sturgeon and salmonids? So Caroline, I'll come to you first about the adaptability, but I'll come to the other two speakers to ask about the vulnerability of sturgeon and salmon to climate change.
Yes, well, the eel has survived for millions of years, freshwater eels. So yes, they are capable of withstanding large changes in temperature. So that they are not among the most vulnerable species related to climate change. So the probably the most, the other threats such as habitat loss and fishing and hydropower are probably the three most important threats to eels.
Thank you. So yeah, Hannah, how is climate change impacting sturgeon?
The main or one of the impacts of climate change will be water temperatures affecting the rivers in which they're able to spawn, because they have a certain temperature range in which they're able to successfully spawn. Luckily for sturgeon, they are roamers and they don't have as high a loyalty to their natal rivers as a fish like a salmon. They do stray to new rivers to spawn sometimes. It's not quite sure what percentage will do that, but it's estimated to be about 5 to 10% around. So it means that they can find new rivers that are more suited to their needs. With climate change, we might see their distributions changing because of these temperature changes. So they are fairly well adapted in that way. But at some point, if there aren't rivers available sort of that meet those needs, then that's when we'll start to see them suffer.
Thank you. And Céline?
So for salmon and sea trouts, they are impacted by the climate change and the rise of the temperature. It was assessed as the major factor for salmon. And so the salmon and sea trout are distributed around Europe as well, but when the population are southern and the temperature are increasing more, then we can see that the drop in the population are a bit more important. So it's getting more and more difficult, but there is as well other factors that we need to consider, like the temperature in the river, but as well like the barriers it has to cross. I think we need to see that as the energy they spend. They will need energy to fight temperatures that are higher than usual, but also the way they have to take, or the energy they have to spend to cross barriers, and it's all things that are costing the fish when they are migrating. So all these things are important for the populations.
Thank you. And on a similar theme, and I think, Caroline, you touched on this briefly in your talk, someone saying that pollution in the UK rivers is currently a significant and sort of very timely problem. Is this impacting eels and other migratory species in their home rivers? So again, Caroline, I'll start with you and then come to the other speakers.
Yes, so as I said, especially the eels that have from the northern part of the distribution, which have a longer life cycle, they accumulate a lot of contaminants. They're very fatty, and fats are typically where the contaminants are stored. And they keep that all their life, because usually a mechanism against pollution for animals is to reproduce, and so release the contaminants through their eggs, or which is not good again for the eggs. So it's a double effect, both on the spawner itself and also on the embryos.
Thanks. Hannah, coming back to you again, is there sort of evidence that the pollution in, obviously we don't have them in the UK, but is pollution an issue for sturgeon across the other parts of its range?
It's an unknown in some respects about what the effects are of certain contaminants on sturgeon and juveniles, and their ability to successfully spawn. So that's something that will develop more as we hopefully start to see these restored populations spawning, but it certainly can affect their survival. There are water quality requirements for the eggs to be able to successfully hatch and sturgeon to be able to grow as needed. So if a river is heavily polluted and dissolved oxygen is very low, then it's very unlikely that sturgeon will be successful in that river. So yeah, it definitely can impact them.
Thank you. And Céline, to you again.
Well, for me, it will be really simple. Sea trouts are very impacted by poor water quality. So we don't have so much sea trout in river with poor water quality. So it's obviously impacting them. But also during their migration, the water quality is also important. If they cross an area where there's a lack of oxygen, then it will be really damaging for the population. And for example, in France, some harbor are dredging, and when it's the migration, and then it's release anoxic mud, and if the fish are crossing these areas, they are just dying almost instantly, and it's impacting, but not only sea trout. So yes, it's important as well.
Thank you. So there's one more question for you each individually, and then if we've got time at the end, I've got a final one, but we'll see where we get to. So one for Hannah: when can we expect to see sturgeon in our UK estuaries? I'm not sure that's an easy one to answer. But,
no, it's not, unfortunately. I guess a hopeful answer would be hopefully soon. We are starting to see them in our coastal waters, not yet in any estuaries, but it is still very infrequent. In the past 30 years, we've had about probably less than 40 sturgeon recorded in UK coastal waters. It's probably underreported because of we haven't done a massive awareness campaign to fishers to report it, but it is still very, very low numbers. So we're hoping that will increase as the fish that have been released continue to grow and age and roam. And I think it's still going to be a while, but I hope to see it happen. And I also hope that in my lifetime we see them back in our rivers. So we'll see.
Thank you. For Caroline, we've got a question. I mean, considering the mysterious life cycle of the eel, do you know if eDNA has been used to sort of try and identify where the European eel exists?
Yes, it has. And interestingly, I think there's also so we can identify European eel, but interestingly, there's I think also been some American eels identified. But the problem with the DNA, the only thing is that we cannot quantify. So we can say the eel is there in that river or system, but we cannot say how many.
One last one for Céline. You suggested that sea trout may have 5 or even more migrations. So does this mean that they've returned to freshwater for spawning and back to sea 5 times over their lives? And if so, are all these populations purely migratory, or are there also non-migratory individuals?
Okay, so I didn't explain fully. Trout can be residents, they can stay in freshwater, and some individuals can be migratory fish. It's the same species, just some are staying in the freshwater and others decide to migrate to increase their growth. But when they are migratory species, migratory fish, then they come back to freshwater to spawn, and they can repeatedly do the migration several times in their life. So yes, in our sampling collection, we had fish that went to sea 6 times or 7, and other ones that was the first migration.
Thank you. So we've probably got about 5 minutes left. So I'm going to wrap up with a question to all the speakers. From what you were saying, it's clear that there's a number of common threats to these species, and also there's the issue of the fact that these migrations are often international. I just wondered whether you knew of any examples where there's sort of opportunities to work across different species to sort of combine efforts, and if not, then where perhaps should we be looking to combine the efforts to protect these diadromous species? That's a big question with probably a very long answer, but any thoughts would be very helpful. Céline, I've seen you've come off mute, so you can go first.
I've got the easy one, so we start like that. It's becoming more difficult for the next one. I think we can combine effort, like Caroline said. There is the barrage, the hydro, how do you say, hydraulic power that are difficult to cross for species. It can be for salmon, for eel, for sea trout, and they are damaging the fish if they cross, as if they manage to cross. So obviously there is some regulation, some effort to follow the migration through these hydropower turbines, and all the fish paths to help the eels and the other species are not entirely the same, but we are trying to mix and to build fish paths that can be helpful for different migratory species. So in freshwater, we are trying to, we can combine our efforts, but obviously when they are at sea, they are not going in the same location, and it's becoming more complicated.
Thank you. I don't know if Caroline, you've come off mute.
Yeah. There's also actually salmon, smolts, and silver eels have very similar physiological mechanisms when they switch from freshwater systems to saltwater systems, and they also display alternate life history strategies or migratory patterns. And certainly I've also picked up information from the scientific literature on smolts for working on eels and when they transition. And I think some collaboration would be interesting to study these alternate life strategies, why an eel chooses not to go in freshwater, for example. And this is a very interesting topic for which we don't know much.
Thank you. Hannah, you're quite at liberty to just say you agree with everyone else, but obviously I don't want to put you on the spot, but please, if you have any thoughts.
My thought to add was just related to the end of my talk about awareness campaigns about records of migratory fish bycatch, and in some species potentially it's important to get information out about restrictions on a certain species. So I know that information can be overwhelming. So for migratory fish in some situations it might be wise to combine efforts and have sort of more aligned messaging and opportunities to share that awareness. So that's just one thing I'd add.
Thank you so much. I'm very conscious that Caroline has to catch a ferry, so I am going to wrap us up on time. So I want to say a huge thank you to all three of our speakers. Fantastic talks and great answers to the questions. And thanks to our audience, especially to those that gave us questions that provided such interesting discussion at the end. I'd like to thank Harriet, who organised all of this evening. So big thanks to her, based in ZSL. So just a couple of last bits. Our next science and conservation event will be in June, looking at the conservation and development projects during crises. So keep an eye on the What's On page of our website for further updates and all upcoming events. Let us know what you think of tonight's event. There's a feedback survey which will pop into the chat. There we go. And yeah, thank you all for coming. And I will say good night to you all. Thanks very much.